Hello, everyone. Welcome to Bea Group's annual showcases. This is our 2024 apprenticeship showcase, and this is designed for emerging bio. I wanted to thank our partner school districts, Downey Unified, Davis Joint Unified School District, and Glendale Unified School District. We are here because of some grant funding, and we're here to work with high school apprentices. We have been collaborating with industry leads, so we're so fortunate to have Karen Ingram here, who is going to tell you about what they've been working on this summer. The apprentices are paid. They have been working. The apprenticeship is for 8 weeks, and this is their showcase to share with everybody what they've been working on. So I'm going to turn it over to Karen, and we'll get started. Thanks for joining us. Karen, take it away.
Yeah, thank you for joining everybody. I see some people that I've invited. I see some guest critics, so I'm very excited about that. This has been a very exciting deep dive. As June said, we had a lot of apprentices in this particular deep dive, and they were all exploring design for emerging bio. Um, the main question that I asked these apprentices is, what goal would they address with emerging biology? And I wanted them to think about the benefits and harms that related to their ideas. So they were in groups of four, and each group was given a specific goal, and the goals all align loosely with Millennium Development Goals. So, we have one group that focused on air quality, one was conservation, one was saving culture. We had two groups that were focused on health, one that was focused on climate change, one that was focused on pollution. Then we had food and pest control. So, nine groups in total with 4 members per group. And a little bit more about the deep dive. They were asked to also consider primary intended impacts and secondary positive and negative and tertiary positive and negative impacts. And I really wanted them to kind of to explore their ideas very deeply and to not fall in love with their ideas and to really analyze them in a critical way. And some of the topic areas that they decided to address were conservation. They wanted to address DNA barcoding. Pollution was biopollution. Pest control against diseases that affect humans. Um, we also had a CRISPR COVID-19 vaccine. We had an alternative cooling system group, a group that addressed Alzheimer's in terms of health. And then we had a group that was focused on fast fashion's contribution to air quality. And we had a group that was invested in the de-extinction of the aurochs. And if you don't know, an auroch is a animal that cows descended from that is now extinct. And then lastly, we had a group that was focused on drought-resistant grains. Um, yeah, and I copied some of the remarks that the apprentices gave in terms of like what the group work was like. So, I think they appreciated working with each other in groups. You can see that from these remarks. And another part of what we did in this deep dive was we created palettes and microbial paintings made from bioengineered yeast. So these are two different types of yeast that have been engineered to produce this like pink or this purple color. So they all created palettes like this. And then I'm gonna stop sharing my screen for a second because hopefully you guys have your paintings with you. If you have them with you, please like hold them up right now. Does anybody have their paintings? Yeah, I see some. Yep. Cool. Emily's is there, Joanne's, Jocelyn, Leslie, Catherine. So yeah, I mean you can see that we did some pretty interesting things. So so let me oops, get back to sorry. Let me get back to sharing my presentation. Um but hopefully they all had fun with let's see. Okay. All right. Okay, so, all right, so yeah, they were also asked to capture data on how their palettes and how their paintings were developing. So it takes like between like 4 and 7 days for yeast to produce its color fully. And so they were asked to take photos morning and night and to collect different types of data. Like for example, this is a screenshot from one of the students or one of the apprentices. They were asked to catalog the conditions like the temperature, the air quality, humidity, whether or not there was rain, because lots of times when we're collecting data around microbes, we want to know if there's been rain. And then you can kind of see based on Destiny's lab notebook, you can see how the colors developed at different rates and how it took about 7 days for her colors to fully develop. And I think the cool thing about this was that different apprentices had their colors developed at different rates and at different brightnesses and saturations. So it was really nice to see the differences there. We could compare. And then these are a few remarks that the apprentices had, yeah, a live lab together via Zoom. And then we had 3 different speakers join us. We had Sabria Stukes from SOSV, which is a venture capital firm that invests in planetary and human health. And she came to see us a few weeks ago and actually stayed about 20 minutes late because she was so interested in talking to the apprentices. So that was pretty cool. And then we had Dee Ardalan, and she is a bioartist that is mostly focused on biomaterials and bioplastics. And we had David Tatzloff from Ayana Bio. And then, I think the student or the apprentices enjoyed having people from these areas speak with them and ask questions. My co-designer for this deep dive is Samika Agarwal, and I appreciate the time that she took in working with all of us. And without further ado, I want to go ahead and start. I think we're going to start with the pest control group. So biopollution. So I'll let you guys take it away from here.
Yeah, okay, I'm gonna be sharing my screen. Just a second.
Thank you.
So here's where we start. Hello, everyone, and welcome to our presentation on the Biopollution Project. We're excited to have you join us today. Whether you're watching live on YouTube, part of the Deep Dive Leader community, or a member of the Bayer Group, or even one of our wonderful and amazing teammates. Thank you for your interest and support in our project. Now we'll be introducing ourselves. First, my name is Xiomara Elisa Rodaz, and I'm really glad to be here.
Hi, my name is Joanne Yu. I'm also very glad to be here.
Hi, I'm Alejo Necochea.
And hi, I'm Madeline Orozco.
Today, we'll be exploring the exciting potential of emerging biology to tackle one of the most pressing issues facing our planet, air pollution. Imagine a world where plants or natural allies could be enhanced to absorb and neutralize harmful pollutants, making our cities cleaner and healthier for all of us. The problem, as we know, is air pollution, caused by industrial emissions, transportation, and other human activities. This poses a significant health risk to animals across the world and contributes to the impending problem of climate change. Despite efforts to reduce emissions, actually air quality remains also a concern in many urban areas worldwide. Our solution is to enter biotechnology, and specifically the idea of developing genetically engineered plants to enhance their natural pollutant absorption abilities. Using techniques such as genetic modification or selective breeding, we can equip plants with the power to actively combat air pollution in our cities.
Here's how we plan to implement our idea. Something like this was already done by MIT Research. They called it the neoplants. The neoplants were genetically engineered houseplants that were altered to be much more effective in cleaning indoor pollution. These three houseplants, the peace lily, the snake plant, and the spider plant, are known for their air purifying qualities. These will be our base plants for our research. We first plan to identify and isolate genes responsible in plants for absorbing pollutants. Then we plan to use genetic engineering tools, CRISPR in particular, to introduce these genes into a suitable plant species. Through these tools, we want to enhance these genes so that they are more effective, focusing our attention to the worldwide air pollution instead of pollution only in the household. Now, next slide, please. Now, what is CRISPR? CRISPR, short for Clustered Regularly Interspaced Short Palindromic Repeats, is a relatively new biotechnology that allows for a faster, cheaper, and a more efficient way to genetically modify organisms in comparison to other technology. Because of time, I will not be getting into the details of how it works. However, the basic version of its process is provided for you in the presentation. It is important to understand the ethical and safety concerns of the public in regards to genetic modification through CRISPR. Next slide. Next, we want to select and grow the modified plants in laboratories, monitoring their effectiveness in absorbing pollutants. Then, by testing and optimizing the plant's pollutant absorbing abilities in controlled environments, we can ensure safety and effectiveness. In this process, we can also see what other effects the genetic alteration causes to guarantee no unintended harmful side effects. Lastly, we plan to scale up the production of genetically engineered plants for real-world applications.
Things to consider. As we explore the exciting possibilities of genetic engineering, it's essential to consider public perception and concerns surrounding GMOs, which are genetically modified organisms like plants, animals, or microorganisms whose genetic material has been altered using genetic engineering. Pest control. Genetic engineering can help create pest-resistant plants, reducing the need for harmful chemical pesticides, which is achieved by introducing genes that produce natural insecticides or make the plant less attractive to pests. The benefits of this approach include reduced pesticide use, leading to a cleaner environment and lower exposure to harmful chemicals for farmers and consumers, and increased crop yields, helping to address food security challenges.
So with all things, there are a myriad of benefits and harms. Some of the benefits of implementing a plant like this is the improved air quality by enhancing the plant's natural ability to absorb pollutants. And then it would also enhance green spaces, creating a greener and healthier urban environment. And this is a more sustainable solution, or in the long term, it reduces the pollutants in the air without introducing new chemicals to it as well. And by implementing this type of plant research, you would also increase the public awareness into the biotechnology and the environmental conservation conversation. But it's important to look at the harms as well. So some of the potential harms are to the ecosystem. One of the unintended consequences on local ecosystems is likely an effect on biodiversity. And there's also a great public skepticism around GMOs or genetically modified organisms, which can hinder the acceptance of a product like this. And of course, this product, while in its development, would need constant maintenance and monitoring to ensure that it's working to its desired effectiveness. But as touched on before, there's also potential pest resistances, leading to new agricultural challenges. But while GMO-based pest control offers significant benefits, maintaining responsibility is crucial. It includes regular pest population monitoring to prevent this resistance by implementing strategies to maintain this biodiversity and ongoing research to refine the pest resistant traits. Next slide, please. So some of the areas that would be affected by implementing these types of plants would be the landscaping industry, by thereby increasing the demand for air purifying plants, with increased incorporation in urban design areas to improve the air quality. There's also an effect on the construction areas by incorporating this into green infrastructure to integrate plants into green walls, rooftops, and other green urban agriculture. So a big thing is that this would hopefully improve public health by improving the air quality, leading to overall better health outcomes for urban areas. And of course, this would affect agriculture, or new techniques for pollution absorbing plants would hopefully reduce the pollution on agricultural lands, promoting healthier crops and ecosystems. And finally, it would affect urban communities most of all, by promoting nature-based solutions with a shift towards natural pollution solutions, fostering an environmental connection and sustainability. Next slide, please. So finally, there's our sources. We gathered a myriad of insights and data from credible sources as listed right there, including the NeoPlant study and the FDA. And there's other sources listed too if you are interested in looking further into this biotechnology. Next slide. And that is it. Thank you for listening to our presentation about harnessing biotechnology to create a healthier and more sustainable future for everyone. Hope you have a nice day.
Fantastic. Thank you so much. I think we have time for one, maybe two questions. Do we have any questions from anybody?
I just wanted to say it's really nice to see you guys focusing on plants and what they can do. A lot of people kind of forget about them. I used to work with flowers a lot. My name's Carrie Havens. I was a molecular biologist back in the day, and now I talk about science. And I actually just wanted to ask the team, have you thought about ways that you could work with nature to kind of mitigate some of those harms that you were talking about, like the biodiversity piece, and how you could incorporate your plants into like the big wig world of plants? Have you thought about connections there?
Well, as I mentioned in my slides, if you remember implementation, we talked about the 3 plants that are more that are known for their filtering: the peace lily, the snake plant, and spider plant. And essentially what we are thinking of is utilizing those natural abilities and finding out what allows them to be so naturally good at it, taking those genes and implementing them into plants everywhere else. So plants that you would typically find outside, to help enhance their abilities in a way.
I see. Thank you.
I have another question, and I want to ask, what surprised you all the most about this whole process?
I wouldn't say that there was anything that was a big surprise in a sense. There were a lot of interesting facts that we learned while studying and researching, especially about the neoplants. Because I guess my Ziamata is the one actually that came up with this idea because of the neoplants. And I guess she wanted to expand on the idea. There was no surprises in any way, but just a lot of interesting facts.
Yes, adding to what Joanne was saying, when they came up with this idea, I actually was surprised about is for us, CRISPR can be seen as something really easy to be modified now in currently times. But the same, it comes with different challenges, as we mentioned, like pest control and like the public skepticism of people. Because even though if we're not going to be consuming those plants because it doesn't have anything to do with food production, but it's actually happened around the world, like in medicine, in like really important things for us to keep surviving every day. I thought of this idea as something that could actually happen.
Thank you guys so much. So we're going to move on. The next group that we have is a group that's going to talk about conservation and DNA barcoding.
Okay, I'm going to share my screen.
Great.
Okay, so hi, my name is Allison. I'm from Warren High School.
Hello, my name is Jocelyn and I'm from Downey High School.
Hi, my name is Camila and I'm from Warren High School.
Hello, my name is Catherine Arcos and I'm from Warren High School.
Okay, so starting off with the table of contents. So for the first slide, we'll go over the importance of elephants. The second one, we'll go over the objectives or motives. The third, the impacts. The fourth, the benefits. 5, the challenges. And 6, other examples that apply to our goal. Okay, so why are elephants so important? Elephants contribute great to their ecosystem and habitat, and they're easier to adapt. Elephants are also the largest living mammals, and they tend to have close family and bonds around their social networks. They're good at maintaining healthy ecosystems and also known as ecosystem engineers due to the fact that they are able to open up vegetation for new seedlings and water or riverbeds. They support numerous amount of species while protecting other animals such as that support their ecosystem services such as catchment, pollination, and social conservation.
So one important concept that our presentation relies on is DNA barcoding. And what DNA barcoding is the method of identifying species using a short sequence of DNA from a specific region of an organism's genome. And by analyzing the DNA of an unknown specimen and comparing it to reference databases of known DNA sequences, scientists can determine the identity of that species. Some important vocabulary that is needed later on in our presentation is the terms keystone species and ecosystem engineered. And a keystone species, some qualities are that they have a huge impact on their ecosystem relative to their population size. They help regulate populations of other species. Hundreds of species depend on keystone species to maintain biodiversity and enhance functionality of their ecosystem. And they're often vulnerable to threats such as habitat loss, overhunting, climate change, and invasive species. Ecosystem engineers are species that significantly influence their environment by creating, modifying, or maintaining habitats which shape the structure and function of their ecosystems. And some examples of ecosystem engineers are beavers, which create dams, and coral polyps, which create coral reefs. So the goal that my group focused on was conservation. And through DNA barcoding, scientists can find poaching hotspots and shut down illegal poaching networks. And how this works is that biologists have extracted DNA from elephants all across Africa by using elephant scat and tissue, and they have genetically mapped out Africa's elephant populations, and this has showed in that each major population of elephants has a specific genetic signature. And by using DNA barcoding on illegal ivory that they have collected, biologists matched this DNA to a certain region they had mapped out, and these locations revealed poaching hotspots to where authorities were sent to catch poachers that were hunting in this area.
So the impacts. Positive impact is that elephant population will be positively impacted and there's a possibility to lessen the risk of extinction due to less death. And some benefits is that the African landscape and wildlife will be benefited since elephants are vital keystone species that help maintain biodiversity and habitat diversity. So the ecosystems affected. Elephants are keystone species and ecosystem engineers. Elephants act as a major agent of seed dispersal, shape ecosystem by producing new habitats, control resource availability, and have many more crucial acts that play a role in keeping their environment in balance. So why is our goal important? Our group loves the environment, the ecosystem, and animals in general. Poaching is an immoral action that has no upsides and only hurts both the ecosystem and the animals itself. With DNA barcoding, we can stop poachers in hotspots, poaching sites. This helps society by lessening the number of crimes committed. With less poaching possibilities come less funds for criminal activity. So the benefits. Who benefits? Law enforcement benefits and anti-poaching rangers. Law enforcement benefits since they use DNA barcoding. And it will be a big aid in tracking down hotspots. And anti-poaching rangers benefit similarly because these rangers have a more successful chance of catching poachers red-handed. And who's interested? Once again, law enforcement and anti-poaching rangers would be interested. Their duties and jobs would be more effective if given access to poacher location. And they would regulate and oversee poaching.
While DNA barcoding does positively impact elephants, there are challenges and negative effects. One of the challenges can be incomplete reference databases of DNA in the region. This increases the chance of incorrectly identifying the species. Additionally, there's the price of DNA barcode. Each ivory is about $100 to examine. Additionally, poachers' main to only income comes from poaching itself, so the poacher's family relies on the poacher's income. These children are able to go to school and live a stable lifestyle due to their father's wealth. While DNA barcoding does help our horned animals, we can also expand to other species such as sharks. So shark DNA is used as evidence to prosecute shark fin poachers. A study in 2015 used genetic markers to show that even in areas where shark fin soup is illegal, the fins may have come from protected and illegally traded animals. Biologists contact law enforcement or lawyers build cases against the individuals that the legal fins were confiscated from. Thank you so much for hearing our presentation.
Thank you, guys. I think we can take the time for just one question, if there's one question that any one has. Any questions? Okay, I just want to make one comment, and that is that I was very happy to see you guys address the challenges with DNA barcoding. Yeah, it's good to be analytical about these things. So thank you for looking into that. Let's move on to the next group. The next group is pest control, and you guys are addressing pest control against diseases.
Okay, so hi, we're the pest control group, and as you can see, our title is Pest Control Against Diseases. So what we plan to do, sorry. So our main focus is to stop pests that cause diseases, and some main pest problems is that they destroy crops, they hurt the environment, they hurt animals, and they cause diseases. So overall, they are not a nuisance, but they are a negative impact to our world, and we want to try and combat that with some uh, with some genetic modifications. So our main motivations to this project were that they inquire a lot of variety and challenges, that we want to help people. Obviously, we want to expand our mindset, so we want to continue growing, and that we plan to have a communal impact, and it kind of help other people, like it kind of goes with it. So one of our plans, well, one of our partners weren't here today. She is going to JOs, but we do have a video to inquire onto the for her part.
Our plan is to genetically modify pests that produce yield and eggs to compromise it. We also want to insert but also like to be more aware of activity and
Next one.
our plan comes from
Emma, would it be possible to have the volume a little bit higher? Thank you. Thank you so much.
What area of benefit is left as hospitalized using and also increasing hospitalization? There are also economic benefits as bills would have to spend, especially when we try to cure illnesses that can be for the family members, and that over having to send money after that will impact their costs. But also because of the as virus, virus and the virus And if less people would get it, then it would be really good. As well as just everyday life. Many people take care of the alcohol and drinking their booze and knowing that everyone is safe and protected from and home pests is very nice to know.
So as you can see from her video that there are many benefits that could to this plan that we hope that we hope has a good outcome into our society.
Hi, my name is Macy, and I'm going to be talking about the harms. So some harms to pest control is that the industries would decline, medical institutions would make like new medicines, people that would study like the diseases may need to change the work due to like things changing, and the public health would need to adjust to the strategies. Some questions and like curiosity questions that we have is, can all pests be impacted or only a certain type with this modification? Can this be done with plants too? And can animals like dogs or cats be impacted with these modifications?
So hi, my name is Carolina, and for the research part of our deep dive goal, pests have been found with genetic change to be successful with this research. And as Monica Guglielmus, a co-senior author of the study and a molecular biologist at the University of Nevada, her and her team have injected new tick embryos and removed the ability to spread Lyme disease in those ticks using CRISPR and the Cas9 system. And studies have shown that plants too can also be modified to keep these pests away. According to Dominic Rizet, hoping I pronounced that right, professor of entomology and extension specialist at NC State, he's modified a plant to produce a chemical to keep these pests away as well. And for pets, we find that they are still impacted by the pets, but possibly they could be problem-free from them. So spreading the information about keeping these pests away, and our method of doing that is by having pamphlets to spread the information in doctors' offices, using social media platforms to further expand the information, and telling friends and family about it to spread the information to people they know as well. And then these would be our sources from from University of Columbia, and also the gene edited ticks, as I talked about earlier. And that is our presentation. Thank you for listening.
Thank you guys. That was awesome. Do we have time for one question? Does anybody have a question?
I have actually a question because I really like the pet piece of this particularly. Have any of you had to actually get a tick off your dogs after walking them outside?
Yeah, like with my old dog, my German Shepherd, she had a couple of them on her like back. So me and my dad had to like get in there with like a pair of gloves and we had to pick him off of her.
Yeah, it's pretty terrible. I just think it would be it would be really nice to not have to worry about that if you go for a hike or something like that.
Thank you for the question.
Okay, cool. Well, we're going to move on. So the next presentation that we have is the CRISPR COVID vaccine in the health category. Take it away.
Okay. Good morning and good afternoon for those who are on the East Coast. Our idea is for our goal health is based on the CRISPR COVID-19 vaccine. And my name is Ceci Lopez and I go to Downey High School.
Hi, my name is Jaylene Guzman and I go to Warren High School.
Hi, my name is Samantha Rodriguez and I go to Downey High School.
Hi, my name is Destiny Martinez and I go to Warren High School.
Main idea. We aim to make more effective and safe vaccines to combat COVID-19 using biotechnology and emerging bio, which led us to CRISPR's DNA vaccine that shows a well-rounded approach to combating COVID-19 by having precise control over antigen design, adaptable to new emerging pathogens, evokes hormonal and cell-mediated immunity, and allows rapid flexibility with ease of manufacture.
Positive impact. The vaccines will have a positive impact on the public, healthcare systems, education, government, and global community will be affected since they rely on one each other. The vaccine will have a positive impact worldwide since COVID-19 pandemic did affect everyone one way or another. Many were scarce of supplies, education worsened, and vaccines helped turn the tables on the pandemic. The vaccines combated COVID-19 and were preventative medicine for everyone, and especially children.
Here are some benefits of the CRISPR vaccine. CRISPR has a long-lasting effect. CRISPR-based diagnostic tests have been approved for emergency use in various countries, providing a rapid and reliable means of detecting COVID-19. It also avoids widespread. People may be interested but may second guess it since it is still new and developing. Lastly, the fears of getting the vaccine may affect their health.
Intended impacts. The primary impact that the vaccine will have will be that they can help prevent diseases like HIV, cancer, and make crops hardier and tackle climate crisis. The secondary impact, the positive of secondary impact will be that the vaccines will have a longer lasting effect on the body, and the negative would be it can interrupt genetic sequences. The tertiary impact will be that it can help prevent diseases like HIV and cancer as previously stated. However, the negative would be that editing genes in embryos may introduce hereditary or heritable changes.
For our vaccine, it comes with many benefits. This CRISPR vaccine can reduce the amount of virus and improve the patient's outcome. This may improve the patient's health overall. On the other hand, there can be harms that can occur. The system can sometimes cut unattended genetic sequences, leading to some side effects or consequences. Since the vaccine is still unknown, these test trials can equal to unknown outcomes. And of course, it is a risk people must be willing to take. Here are some of the resources we used to put this together, especially our information on CRISPR.
That is it. Thank you all. If you have any questions, feel free to ask, or you can also email us.
Great. I love that you put your email addresses on the end of this presentation. We have time for one question. Does anybody have a question?
I do have a question. So during the pandemic, was this one of like the top candidates to be chosen as the COVID-19 vaccine? Like, why did they choose mRNA vaccines instead of CRISPR?
Kira?
I can help with that. Yeah. So what CRISPR does is it writes inside the living cell, right? It changes the information inside the cell itself. So you guys were talking about a lot of the ways CRISPR can do a lot of different things, right? And what that means is that the scientist has to pick the target and then write the little piece of DNA and send it into the cell. But the way your immune system works is that it kind of trains itself on the environment around it. It has some cells that combine and recombine and end up with sensors, little receptors that can identify things that might be harmful and say, nope, we're not gonna deal with that. So what the mRNA vaccine does is it shows up. One, we know how to write that information relatively well. Like we're pretty fluent in that particular language. But two, it shows up with the mRNA vaccine, uses mRNA to teach your existing immune system what to look at. So it produces like a cardboard cutout of the virus and kind of like waves it around, and your immune system says, oh, that's a bad thing, I'm gonna get it next time. CRISPR vaccines are, you would use that to deliver a new function to your cells. So you would use that to make the sensor that could recognize COVID-19. Or maybe if you got really creative with it, you'd use it to cut out pieces of the virus that were stuck in someone with long COVID, right? And the truth is, we're just not quite that good yet. We don't have all the skills we need to actually make that real. So the mRNA vaccine had advantages in that it was fast, in that we knew how to do it, and in that it kind of worked with our body's existing immune system to train it to get the right thing and help keep us safe.
So that's how.
Yeah. Mm-hmm. Sorry, I was just going to say one more thing.
No, I mean, that's great. That's how cutting edge you guys are.
Mm-hmm.
But I want to
Yep.
just give a shout out to Kira. She is a science communicator and a scientist that works at Pivot Bio. And I'll put the link to Pivot Bio in the chat in a minute. But I asked her to come because I knew she would give you guys wonderful questions and insights. So thank you for being here, Kira.
Yeah, for sure. And unfortunately, I'm going to have to leave a little bit early, but you know what? I will just put my email in the chat. So if you guys have questions or are curious or something like that, feel free to reach out. And I'm always happy to chat about this stuff.
Thank you so much.
Yeah.
So the next group that we have is an alternative cooling system. And this is the group that is addressing climate change. So I'll let you guys take it away.
Hi, our group is about climate change. Can you guys hear me by any chance?
You're a little quiet.
Kevin, can you talk about the intro so I can fix my mic?
Yeah, sure. So our group's based on climate change, and our subject is creating new cooling systems. Hi, my name's Kevin.
Hi, my name's
Hi, my name's Ian.
I'm Caitlin.
Okay. Our main objective is to reduce natural gases and climate change in like offices, apartments, schools, hotels, like mostly buildings, by creating a new cooling system that relies on enzymatic reactions and enzymes. It's like the picture here is just mostly like new systems that are used right now, like older systems I meant, and like the design they use. Okay, so enzymes are molecules, typically proteins, that are used for biochemical processes. And an enzyme reaction, it's like enzymes bind to substrates that create a conformal change, and then a biochemical reaction, it then happens after. So we want to use those reactions to create a reaction that absorbs heat in the surrounding environment to keep like the rooms and places at like a constant temperature. So we want to find enzymes that can create endothermic reactions.
Who's harmed? Developing our idea, we do not know the actual enzyme that we're going to use, but we strive to create an enzymatic reaction that is eco-friendly, non-toxic, and non-harmful to the ozone layer. With the business aspect, potential companies that can be harmed could be Emerson, Whirlpool Corp, Carrier Global Corp, and etc. This is because HVAC companies use chlorofluorocarbons and hydrochlorofluorocarbons, which are harmful to the ozone layer. These carbons are made up of carbon, hydrogen, chlorine, and fluorine. And so yes, these are really known for being energy efficient and being like less toxic. And so these are very good options. But as a team, we we seek to find a new way to minimize the damage to the ozone layer and with our new cooling system. And with these images, it just shows how the CFCs or the chlorofluorocarbons are emitted into the air and then broken down, and how it depletes the ozone layer. And although some may find this a good idea, this idea hasn't been completed or published, so we still don't know if this idea could be costly or even efficient.
So when it comes to who we believe will be benefited from our idea, the goal is that it benefits every living organism, so ranging from algae to humans, bacteria, animals, or plants. Specifically in this presentation, we're aiming at looking at who it would benefit as humans. So specifically, people in heat-prone areas would be given a new cooling source, providing for comfier living conditions and situations. Thus, this would help improve humans' daily lives by reducing temperature of ceilings, walls, and furniture, like furnishing. And this would help exceed the limits of the temperature rising in the homes. As well, those living in urban areas would benefit chronically from our idea because the urban heat island effect, which by definition basically means an increase in energy costs like air conditioning and air pollutant levels for increasing, which would also lead to heat-related illnesses. This would all be reduced by our idea of cooling systems. Among other variables as well, it would also improve air quality and circulate better and cleaner air and remove harmful pollutants from impacting the air. As well, it'll also be more energy efficient and the cost would be much better. So thus, our cooling system would create a healthier environment and mitigate the excess of harmful pollutants in the air. And then also that image on the bottom right just shows how our cooling system could help impact the lives of people in their daily lives at homes.
Our approach. We plan on addressing both the harms and benefits by identifying the problems that might come with the cooling system and gathering data about it and information. And we also list the harms and benefits that come from the cooling system, direct and indirect, and analyze the impact on the environment and consider external factors and consult with stakeholders. Positive and negative effects. Some of the positive effects include improved air quality because it'll help reduce the greenhouse gases, and it also helps to preserve food. It gives it an extended shelf life, and it uses less energy, and it has a decreased reliance on natural resources. Some negative effects are that it can have a high initial cost, and the traditional cooling system manufacturers and workers might face economic challenges with the shift towards the new cooling system, and new technology could fail and lead to dissatisfaction with others.
And these are the sources that we use, and we all incorporated these sources into our texts, and yeah. And thank you all for listening and giving us your undivided attention. And if you guys do have any questions, you guys can just give us a shout, and we'll be happy to answer your question.
We can take one question if anybody's got a question they'd like to ask.
Yeah, I think it's a really creative use of enzymes. Enzymes are very cool little machines that can do almost anything. So I'm curious, which enzyme did you read about that had you learning that they can like take in energy to perform work? What was the inspiration, I guess?
There wasn't like actually like an enzyme that was specifically like labeled. It was like mostly in general, like people found like certain ones that if they more like changed it a bit, they can do that. But we couldn't find like a specific name for one.
Yeah, it's new to me too. So I was curious. I would actually wanted to go look them up if there was one out there. Okay, I think that's the last one for me, folks. Thank you so much for sharing your projects and everything. It's really great to meet you, and please do reach out. Thanks, Karen.
Thanks, Kira.
Thank you for.
Take care.
So we have our next group, which is another health group that's focused on Alzheimer's projects. You guys ready?
Yes, I believe we're ready. I think somebody's gonna share the screen. Let me see if I can pull it up. All right. Let me just share my screen. And then all right. All right. Are you able to see it?
Yes.
Yes, thank you.
Let's see if we can present it in a way. All right. I think that's all right. So good morning, everybody. My name is Ben, and our goal was for the health group, and our topic was Alzheimer's. I don't know if the rest of my group is going to introduce themselves.
Hi, my name is Monse, and I'm from Warren High School, and I'm also part of this group.
Hi, my name is Melanie Mesa, and I'm from Downey High School, and I'm a part of this group as well.
And then I don't believe Jesse's here. We're not really sure why, but he's not here yet. But moving on. So we're gonna start off with what Alzheimer's is. So Alzheimer's is a type of brain disorder that causes problems with memory, thinking, and behavior, and it's a gradual and progressive condition. The symptoms usually grow severe enough to interfere with daily tasks, and this disease can be fatal. The causes, the main cause is a buildup of two proteins. The beta amyloid protein, which is involved in Alzheimer's, is formed in the breakdown of a larger protein called the amyloid precursor, which comes in several different molecular forms that collects between neurons. The beta amyloid 42 form is thought to be the most toxic form. In the Alzheimer's brain, abnormal levels of this naturally occurring protein clumps together to form plaques that disrupt the cell function. And the other protein is called tau, which forms in neurofibrillary tangles, which are abnormal accumulations of the protein. The healthy neurons are supported internally in part by structures called microtubules, which help guide nutrients and molecules. Which help guide nutrients and molecules from the cell body to the axon and dendrites. In a healthy neuron, the tau normally binds and stabilizes the microtubules, but in the Alzheimer's disease, abnormal chemical changes cause the tau to detach from the microtubules and stick to other tau molecules, which form threads and eventually join together and tangle inside the neurons, which block the neuron's transport system and harms the synaptic communication between the neurons. Our solution, the treatment for Alzheimer's disease would minimize or remove the buildup of these proteins around the brain cells and will come to life through trial and error, testing out what works and what doesn't, and by doing further research into the specifics of the brain. Since the brain is a vital and fragile organ, research must be done in order to completely understand it so that the treatment can be most effective. And to be more specific, is our next slide. Montserrat.
There have been studies into the disease and synthetic alpha sheet peptides, each made up of 23 amino acids, which could be part of the solution. These peptides neutralize the amyloid beta oligomers in human neural cell cultures that cause Alzheimer's. So our solution would be synthesizing these amyloid beta oligomers and find out a way to insert these into a patient's brain via brain surgery. The benefits of a treatment like this would reach elderly and those who currently have Alzheimer's, since it would decrease and prevent the buildup of the proteins that cause Alzheimer's. This would also help the family of the Alzheimer's patients because they won't have to go through the heartbreaking process of watching their loved ones suffer from the neurological disease. Those who benefit would be interested in this treatment because even though our treatment might be expensive or it is still experimental, if it does work, they would have a chance of having a normal life again because the symptoms of Alzheimer's can be life-changing, such as the symptoms they go through are memory loss, poor judgment, repeating questions, and loss of initiative.
Okay, so who is harmed and how do we address the problems? Those that are involved in the human trial could be harmed, as well as those who have a bad reaction to the drug. We could address the issue of those that might be harmed by deducting the reasons until we can pinpoint what exactly causes this harm, and then we can address on a case-by-case basis. We could also use the information previously mentioned to put warnings on the medication as to minimize the bad reactions. So what challenges would you encounter in achieving our idea? So the biggest challenge right now would be coming up with a medicine that would work for this, and there would likely need to separate medications for the different proteins targeted, Another problem would be that there hasn't been enough research into the disease, leaving many holes where a solution could be found. And another challenge would be finding a company to fund this project because it is likely to take many years to finalize the drug, and it is a risky financial investment. And this was our source page, and that was it for our presentation.
Thank you guys so much. That was super interesting. Do we have a question? Okay, we're going to move on, but if you guys have any questions, please feel free to put them in the chat. The next group that we are going to see present is a group that's focused on saving culture, and they have elected to make their project about saving the auroch. And an auroch is a animal that cows descended from, so modern domesticated cows. So I'll let you guys take it away.
Hello and good morning, everyone. Our group's goal was on saving culture, and this is our presentation on the de-extinction of the aurochs. To introduce ourselves, I'm Alyssa Sandoval.
I'm Jackson Sabatino.
I'm Caitlin Baron.
And I'm Yasun Ortega.
Throughout history, various cultures have seen the oryx as a symbol of strength and resilience. The oryx were frequently depicted in cave paintings such as the Lascaux and Chauvet caves, indicating their importance in the people's lives back then. For example, a specific group of people whose culture the oryx were involved in and around were the Aurignacian people. They depicted the oryx along with many other animals at the time that were important to them in the Chauvet caves as a symbolic and religious figure, as a symbol of strength and just the need to keep on going. Our idea to save the cultural impacts of the oryx is to use gene splicing technology to help combine certain genes together to hopefully resemble the unique traits that an oryx has. For that, we would be using selective breeding to separate the unwanted traits from the wanted traits that we're going to use for our later splicing process. Now, to find these traits, we would need to take them from the genetic coding of a current living ancestor of the oryx, which in this case would be common day cattle, certain types of mountain cows, and even a certain type of ox. We would breed this genetically modified cattle with these specific traits to create an genetically modified offspring that would most accurately fit the description of an oryx as we're scientifically able to. Now, me and my team think this de-extinction process would have a positive impact on cultures who idolize the oryx, like the ones previously mentioned, and would help rekindle their culture that has been long gone due to either assimilation, time, or any third-party reason. The process would also positively affect the biodiversity epidemic that we are facing in the world nowadays, and would help reestablish the unique biodiversity in the parts of the world that the oryx live in, and would help that part of the world recover from the increasing biodiversity loss epidemic. We are aware about the concern that cost brings up because of all the use of genetically modified weapons, genetically modified splicing, cattle. So that's why we plan to use the most cost-effective materials and most cost-effective and safe cattle for the splicing and breeding process to secure any cost issues and any breeding issues. And overall, this will just as a whole help push the idea of de-extinction into mainstream to help revive hundreds of thousands of animals, revive hundreds of dead and lost cultures, and continuing the work done by companies such as Colossal Laboratory and Biosciences, who, like us, specialize in the de-extinction process and the saving of culture.
So some benefits to this are it allows
cultural groups to preserve and maintain their history and culture. It can also address habit loss issues and positively influence biodiversity in Europe. It can also both ancient civilizations in India and the Fertile Crescent depended on aurochs to develop their resources. Aurochs grazing and trampling disrupt the soil, creating conditions that allow pioneer plants to establish themselves. There are also some harms to this, which include existing animals could be harmed due to aurochs taking away resources for animals on land. Unknown rare diseases could be intentionally selected for breeding and passed to the recreated species. Caution is needed to avoid harming existing ecosystems, especially by watching for interspecific hybridization that could cause the genetic extinction of current animals. Assessing community support and the effects on local culture, economic, and health is also important. And then also common fears of predatory attacks, nature-based tourism offers business opportunities for local.
Our main goal is to revitalize extinct species. Some impacts following de-extinction would be preserving biodiversity, but when adding a new animal to an environment, it could possibly cause difficulties for other species to adapt to change, and it can be harmful in some cases. However, it's still hypothetical, which is why this technology would need a lot of testing in a control area. In terms of society, bringing back certain species can preserve cultures that once heavily depended on that particular animal and continue to promote diversity within culture. Yet many disagree with the goal, believing that bringing back an animal that has already gone extinct is immoral or it goes against religious beliefs.
In addressing the benefits, cultural groups would be interested, as this idea will bring recognition and an effort to preserve past and existing cultures from being lost in history. In addressing the harms, that would include ensuring resources are not taken away from existing animals. Instead, this process of de-extinction is used to help animals on the brink of extinction, and also including the people and communities in this process to ensure that there's support and people are willing to coexist with aurochs. And this could be done by inviting communities to a gathering that we would hold, presenting our idea to them, explaining the benefit and how this idea would help to improve their lives, and also listening to the responses and opinions on this idea and gathering their feedback. And these are our resources and our contact info. Thank you for your time.
Thank you guys. That was wonderful. Really liked how you delved into the benefits and harms. Does anybody have any questions or thoughts before we move on?
I'm just wondering, what do you guys personally think? Would you guys like to use it or not for the purpose?
I think so. It's a cool idea. It's also just unfathomably like something that you would never imagine we would ever have the possibility to do. So the fact that we can do it and we can prevent certain species from dying, it's just a really cool idea. Now, I do get the ethical concerns and the religious concerns, but I still think it's worth doing because a lot of great ideas nowadays come from big religious concerns, like the whole thing with airplanes. People have concerns about you weren't allowed to fly, so why are you doing it anyways? But I think it's really cool. So I think that, yeah, definitely.
Okay, cool. All right, well, we're going to move on. And our next topic is air quality and fast fashion.
Hi, my name is Jocelyn, and unfortunately, my other teammates couldn't be here, but I'm gonna do the presentation. Let me just share my screen.
Thank you.
Can you guys see it or no? Yeah?
Yep, we can see it.
Okay. Is it working? Okay.
We can see.
Okay.
It's good now.
We're gonna we did our project on how fast fashion contributes to poor air quality. And so we wanted to create sustainable fabric that will double as being biodegradable. So we decided to use algae as our fabric, and we wanted to use algae. I don't know how to say that. Sorry, because it's biodegradable, it's allergen, it's non-toxic and allergen-free. It requires less energy and resources, and no pesticides are needed for it to grow, and pesticides are bad for the environment. So overall, we thought that this was the best material that we could use. And algae improves air quality because it helps purify the air by removing carbon dioxide through photosynthesis. And it uses other materials that aren't biodegradable using other materials that aren't biodegradable release toxic greenhouse gases into the atmosphere, which this pollutes the air. As I said before, algae doesn't require pesticides, and fast fashion contributes to 10% of carbon emissions, but using this material, we can change that. Challenges that would be faced using this material is not everyone will feel comfortable wearing algae as a fabric, and the company will need to use a vast amount of land as you need to grow the algae, and may have a shorter lifespan than a piece of clothing with other materials because it is biodegradable. The intended impact is that with developing this new fabric, we intend to better the air quality. And since this fabric is biodegradable, it will eliminate the landfill process, which emits particulate matter along with carbon dioxide and methane into the atmosphere. And the algae itself will also be removing carbon dioxide through the process of, like I said before, through the process of photosynthesis. And that's our presentation. Let me stop sharing my screen.
Thank you, Jocelyn. Does anybody have any questions?
I have a question. Are there any like manufacturers that you know of now that are kind of using this technology, or there's nobody out there?
I think that there is people trying to use this material. I think I've heard of a company, but I'm not sure what it's called. But I think there is some developing companies trying to do it.
Thank you.
Yeah.
Okay. We are going to move on then. So the final team is the drought resistance grains team. So I'll let you guys take it away.
So good morning. We are the drought resistant grains. To introduce ourselves, my name is Emily Partita.
Hi, I'm Adrian Perez.
Hi, I'm Brandon Castro.
And our fourth partner, unfortunately, is not here right now. His name is Kevin.
So we're group 7, and we're exploring biotechnology within food. We are going, so you might ask yourself, you might ask yourself, why do we need to explore food technology? Specifically, why would we need drought-resistant grains? Due to the rapid population growth and the Earth's change due to climate change, drought-resistant grains can assure safety and a reliable choice of food for those affected by poor climate and rural areas. So our goal is, while the population grows, food supply does not. We are hoping this will guarantee a delay in the growing food crisis. This will provide a stable availability of food supply. These drought-resistant grains will thrive when being fed less water than the average grain. They will thrive in rural areas and poor air conditions. This drought-resistant grain will require certain nutrients to remain beneficial to humans, but with the right nutrients, it is possible.
You may ask, why do we need drought-resistant grains? Well, the Earth's population grows at an exponential rate, while our food grows at a linear rate. It's called Malthusian theorem. Um, there will be a moment in our future of our lives where we will have insufficient food um, in proportion to the population. So we won't have enough food available for the amount of people that are being born. And drought-resistant grains can help this because we'll have more food available and we'll be able to feed more mouths and keep more people alive.
So for our benefits and harms, our benefits can have a positive impact on communities that are in rural areas as they will suffer malnourishment due to them not being able to have the correct weather conditions to grow regular and average grains. The reduction in malnutrition will occur and the communities will be able to farm in their day-to-day life with an easier form of grain. Since less water is required to farm these crops, they can save money and they can save transportation costs of food from receiving it from other areas that will be able to grow their grains as they have better weather conditions. And the healthcare will be reduced due to their lack of malnutrition with the new drought-resistant grain. The harms will be existing farmers can suffer um, um, a reduction in their average farming life. They have their tactics and their strategies, and it will be sorry it will be it'll be
Yeah, I can talk about the
yeah.
harms. Okay. A lot of farmers who provide food for those areas will lose a lot of trade and commerce. As you know, people will not be looking to buy food from external sources, they'll buy from internally. So external farmers who provide food and provisions for that area will lose a lot of opportunities to sell their crops. So that might affect them. Other effects is that, it's unknown what's going to happen when you introduce a new crop into a new ecosystem. So there's harms that you have to take into account from that. And other harms is that as you're introducing this new crop, you have to worry about overcultivation of the land. And if you overcultivate the land, then eventually the land will lose its nutrients and unable to grow the crops, which will cause an even bigger problem with the food supply going down and down.
I was gonna say, I think the screen is frozen.
Is it?
Yeah.
Okay, let me Can you see it now?
It's loading.
Yeah.
We still
If you
can't.
want, you can share the link and I'll share the screen.
Okay.
Okay, thank you. What slide are you guys in?
We're on slide 5.
And the benefits and harms, right?
Yeah, slide 4.
So like I was saying, the overcultivation of crops, if the farmers grow too much crops, eventually the soil will lose its nutrients and they will be unable to grow these crops, and they need to learn the practices of sustainable farming. And there's also, introducing these genetically modified crops is also you have to convince the populace that these crops are able to be consumed. Like Karen, she was talking to us about golden rice. That is a crop that has nutrients and vitamins in it, and it took a while for that crop to be accepted by the general public. So that's one thing we'd have to work on, is just getting the general public to accept our crop.
Okay, go to the next slide. So some impacts are increased yield of the crops of grains, because they can be produced even in dry conditions, and it reduces the risk of famines. Another positive impact is increased income. So farmers, since they can have higher yields, they can make more money off of the drought-resistant grains. And then another positive impact is water conservation. Since the grains use less water to be produced, it'll help us conserve our water. So we have more water for drinking or showering or more crops. And these are also resilient to climate change, since they can adapt to weather patterns, and it ensures a continuous food supply. Some of the negative impacts are it can disrupt ecosystems, because the introduction of a new crop can affect local ecosystems and wildlife. And it'll affect everything around it, since it's not hasn't been there before and it's new. And then also the initial costs of the grain can be a barrier for small farmers, since they don't have that much money. So it could be expensive for them. Also, genetic modification can raise environmental and health concerns. There's more research that's needed to be done. So we need to figure out more of the health concerns and what this can do to the environment in the future. And then finally, pesticide and herbicide use can require more like chemicals, and then it can impact the environment and human health as well.
Just to conclude, that drought-resistant grains will ensure that there's a steady food supply for a while. However, it's still not a permanent solution, as eventually exponential graphs always overtake linear graphs. So we would always have to find more solutions to produce more crops and food. But this would be a nice way to put a little jump and provide more food and nutrients for those communities that are malnourished and need more crops and food. And by working together, we can create a stronger, more sustainable future for agriculture. Next slide, please. Thank you. And if you guys have any questions or want to contact us, our emails are right there.
Wonderful. Thank you guys. So that pretty much does it for all the presentations and all the group work. Does anybody have any thoughts or questions at this point?
I think it's just a personal question, and what surprised you the most about yourself? Or what did you discover about yourself during this apprenticeship?
And I guess that's open to any of the apprentices.
I discovered that like teamwork is really fun, and I enjoy working with the group and collaborating with my teammates and my coworkers and my partners.
Yeah, along with what he said, I realized how important it is to communicate and how important it is to have a steady form of communication and not take gaps in between when you're working with other people.
Just wanted to jump in, team. We have a video from an apprentice that we are also showcasing today. Aleda, if you are here, can you please share your screen as well?
Yes, I can do that.
Just want to make sure we have this sent and shared as part of this apprenticeship showcase. Thank you.
Give me a second, sorry.
And I just want to thank everyone. And yeah, look, like that feedback is super helpful and important to work with teammates.
Okay, please let me know if you can hear the sound.
Can you please make this in full screen as well later? Thank you.
Well, my name is Jesse Barrientos, and we're going to be talking about Alzheimer's disease.
Can you hear that?
Yes, we can
Yeah, you
hear
can
it. We don't see it just yet.
Okay.
Can everybody see it okay?
Just give it once she tries to go full screen. No.
Oh, you can't see it when it's full screen? Okay.
First of all, what is Alzheimer's? Alzheimer's is a type of brain disorder that causes problems with memory, thinking, and behavior. This is a gradually progressive condition. Symptoms eventually grow severe enough to interfere with daily tasks, and in the end, the disease is fatal. So, our solution. Our treatment for Alzheimer's disease would minimize or remove buildup of proteins around the brain cells. Will come to life through trial and error, testing out what works and what doesn't work, and by doing further research into the specifics of the brain. Since the brain is a vital and fragile organ, research must be done in order to completely understand it so that the treatment can be most effective.
Thank you.
Thank
We
you,
just want to
Jesse.
make sure that he got an opportunity to present.
Thank you, Leda. Brilliant. So yes, do we have any other questions for the team and for the group? Checking the chat. Okay. Well, thank you so much, team, and congratulations for live streaming your showcases today, apprentices. All of your presentations have been fantastic. Thank you for, your efforts, your input, and your energy today. And it's, great to see what everybody has been up to in the past couple of weeks. And a big thank you to Karen as well, the deep dive lead for Design for Emerging Bio in this session. And before we close out, I do want to let everybody know we have one more live stream showcase this afternoon at 3 o'clock for business development and entrepreneurial thinking. And we also have 4 other virtual showcases tomorrow on Thursday. So we have Arts, Media and Entertainment Collaborative at 9 o'clock, 9 to 10:30 a.m. Then we have Next Generation CRISPR from 11 to 1 o'clock and Generative Artificial Intelligence from 1:30 to 2:30 p.m. And last but not least, we've got Product Service Innovation Development from 3 to 4 o'clock. So you will see all of that in a schedule on our social channels, Bayer Group, on Instagram and on LinkedIn. And of course, you can also reach out to us at any point if you have any other questions from these presentations today. So thank you, team. I'm going to close out on the live stream right now. And if everybody can stay on Zoom so we can run through just the final next steps. In fact, I think this has stopped live streaming already. Is that correct? Can you see Juan and Barbie on our YouTube?
No, it's
still live streaming. Okay, hang on just a minute. Thank you, team. Bear with me.